Perfect. Well, good morning. Thank you for being
Good
here.
morning. Yeah.
Okay, it is my pleasure to have you all here. Thank you for coming today. And thanks to the folks online for showing your support for Allison. So a little bit of an introduction for Allison. Allison graduated with a Bachelor of Science in Marine Bio from URI back in 2014. And before she came here, she did a lot of different things. She was a groundfish observer for a little while up in Alaska. She did some field work over on Guana Island at BVI. In her time here, she has done some pretty amazing things. She was an NSF Strong Coasts Fellow. While she was here, she was also a graduate student rep, as you all know. But she also had some honors that you might not know about. In 2021, she was selected as a Coastal and Estuarine Research Federation's Rising Tides Fellow, and has contributed onto that program and that professional society. And last year, and finishing up right about, I think this month, she was selected as a 2022 and 2023 American Geophysical Union's Voices for Science Fellow as well. She's helped us tremendously in growing the social media, literally, for GROW. So if you haven't checked it out, you're in the dark ages, because she's doing amazing things over there and sharing the work that we're doing, including creating a video or a PSA that was been showing like the past month over at Caribbean Cinemas. So if you hadn't had a chance to check that out, we can also share that. But I'd like to just compliment that Allison has just been a positive force in our lab while she's been here. Her positive and collaborative attitude has really made a lot of things work well, including her mentoring of students, young and old, and her tremendous work ethic. So today she's going to be sharing her thesis work, which is focused on looking at some different grow out options at the nursery. And I'm really excited for her to share with you all what she did and what she found. So thanks. So just as a reminder too, I know we're in thesis season here, going strong in thesis defense. And for those in the room, I'm pausing because we just had an airplane go by. But just as a reminder, Allison will speak for probably about 45 minutes or so. There'll be time for questions from the general audience, and then we'll go into a closed door session with just her committee. So for the committee members, I ask you to just hold your questions till that time. We'll let the public questions come first. Okay, thank you.
Yeah. Good morning, everyone. Thank you so much for being here in person and online. Hey. A special thanks to my committee members, Kristen and Marilyn, for coming and being here after such a long trip from Guam. Wow. And Ken, I know you're in Louisiana, but hey, that's far too. So thanks for being here. I really appreciate all the support. And I have to do the thing. Okay, there we go. I really appreciate all of your support as we have, as Kristen mentioned, a long marathon of thesis defenses ahead for us over the next 2 weeks. So to keep things exciting, to get you all jazzed and to spice things up a little bit, we have our friends and musical genre spanning, record breaking musical princess, Miley Cyrus, here with us today to help me along as I talk through some of our results and how I used different clumping strategies and mangrove substrates to grow mangroves, specifically red mangroves, in our mangrove nursery. And you're probably thinking, why Miley Cyrus? Well, why not? You'll be happy to know that Miley Cyrus and mangroves actually have a few things in common. Although Miley is a human and mangroves are trees, both Miley and mangroves are a salt-tolerant species. So Miley has her liver to thank for her ability to handle salty situations, but mangroves have special adaptations that allow them to actively excrete or exclude salt from the plant. Furthermore, mangroves are extra unique in that the propagules, which are the mangrove seeds, start to grow on the parent tree right away, which is very interesting. And last but not least, mangroves and Miley are considered extremely resilient, able to bounce back from severe disturbances. Now, as I'm sure Miley herself would tell you if she could, the ecosystem benefits mangroves provide to coastal communities, especially to communities like those on islands here in the U.S. Virgin Islands or USVI, are really, really great and important. For one thing, our coastlines are protected from wind and wave energy from storms, thanks to mangroves. You know what else? Our coastlines are protected from erosion, thanks to mangroves. In addition, I know, just a cute little erosion guy there. Are you looking for an eco warrior to help battle our climate change impacts? Then look no further than a mangrove, because mangroves actually store large amounts of atmospheric carbon within their soils. Those fish you love to eat at Fish Bar, yep, they grew up in the mangroves. And besides providing essential habitat to a variety of fish, bird, and other species, they provide us places to explore and learn to love. I mean, look at Miley. She's never looked happier in the mangroves, right? Plus, with the use of her private jet, you and Miley can explore mangrove habitats around coastlines in the world in tropical and subtropical latitudes, including the mangroves here in the US Virgin Islands. Here in the territory, we have 3 islands, St. Thomas, St. John, and St. Croix. And around the territory, you can find 3 of the 80 global mangrove species, which include the red, the black, and the white mangrove. Unfortunately, wrecking balls of chronic deforestation, pollution, and sea level rise all threaten the viability of our mangrove systems and the ecosystem benefits that they provide. But never fear, because mangrove restoration is certainly here. Loosely and for our purposes, mangrove restoration is the establishment or return of a mangrove habitat to an agreed upon state of health. In an ideal situation, mangroves would always be able to bounce back from disturbances. However, some of those stressors we just mentioned can make that hard. So one technique we can use to help recover our mangrove forests is restoration planting. Since mangrove forests here in the USVI have been slow to recover since the 2017 hurricanes Irma and Maria and continued deforestation, our wonderful team has been working diligently over the last few years to determine where restoration is needed as well as feasible. And in an effort to aid all of the restoration that's happening, we've grown a mangrove nursery. Yay! Now, the species most likely to be used in a mangrove nursery as well as restoration planting is the red mangrove. And that's because red mangroves are some of the hardiest. From our own observations and personal communications with other restoration practitioners, red mangroves can grow in almost anything: clay, mud, manure, seawater. And this is really important because mangrove nurseries on island communities face challenges, such as the importing and buying of goods, such as potting soil, the price of those goods, and last but not least, the landfill in the room, waste management. Long term, it is not always feasible for small communities to continually buy potting soil, as that is a really expensive cost. Nor is it always feasible to match the nursery conditions to those of the potential restoration sites, right? There's no way you could climb in and out of here in this picture through those mangroves carrying buckets full of soil to the nursery. Most restoration sites are not easily accessible by shore. And furthermore, the work to do the time and human power needed to do that kind of work is extremely costly. And last but not least, special permits would be needed. So as we've grown our mangrove nursery and walked through all these different restoration things and how we can help the territory, our team has been thinking about these challenges. And that's where my project comes in, where I tried to maximize plant growth while incorporating some sustainability practices into our nursery. And to begin, I asked myself a few questions and things that I wanted to figure out as I went through this scientific journey. First, how do different soil and crushed glass substrates affect plant growth? If mangroves can virtually grow in anything, why not crushed glass? As this is a win-win for everyone, right? It keeps repurposable waste out of landfill, and over time, it cuts down on our need to buy potting soil. Second, how do clumping strategies affect mangrove growth in a nursery? And when I say clumping, I mean the adding of more than one propagule together in a pot, right? The clumping of individuals. And in my thesis, which you'll see later, I also consider a clumping treatment a propagule grown with a wooden control dowel. But we'll get that more later, just keep that in your mind. For some coastal wetland species, we know that when these plants are clumped together, there are biological benefits to clumping, such as sharing oxygen at the root level in stressful times. But for mangroves, we don't know if these same biological benefits exist in the field or in the nursery. In addition, are there physical benefits to being planted next to each other in the nursery? I don't know. We're going to find out. Third, what treatment and site combinations maximize propagule growth? This is extremely important to us as the mangrove nursery team because we believe that the bigger and healthier the plants are now, the better they'll be able to establish and survive transplant shock later on in the field. And last but not least, how do treatments affect plant morphology below ground? So sure, a plant can be tall with lots of leaves, but that tells us nothing about what's going on in the substrate, right? What different types of roots are there? How is the plant's biomass affected by these treatments? My project had a soft start in October of 2021 when my wonderful girlfriend Jenna Sheremy and I collected over 3,000 glass bottles from restaurants here in St. Thomas, saving them from going to the landfill. We crushed them in vine masses of the Virgin Island Marine Advisory Services glass crushers. So thank you, Howard Forbes, if you're online, for letting us use that. And as you can see, cupped in my hands here is an example of what that glass substrate looked like from bottle through the crushing machine, bam, final product. But the real work began in May of 2022 when we collected red mangrove propagules from 4 sites within the territory. On St. Thomas, we collected mangroves from Perseverance Bay, St. Thomas East End Reserves, or STEER, and Oasis Cove Marina. And on St. John, we collected propagules from Annaberg Bay. Now, a quick note here, Annaberg Bay was actually added as an opportunistic collection site because during collection, we found that propagule drop timing, or when trees release their propagules when trees release their propagules and they fall on the ground, this differed from what we had seen the previous year from site to site. So we were actually scrambling, trying to find propagules we could collect from. And this is important because as mangroves are protected in the USDI, our permits only allow us to collect propagules from the ground. In the end though, we collected 480 propagules, 120 from each site. And we only collected the best of the best. So no damage, no discoloration, no herbivory, because obviously our project deserves the best. Come on. After being collected, propagules were kept in buckets of seawater for no longer than 48 hours until processing and intake and planting. At the time of processing and intake, we collected a few different metrics on these propagules, including total length, mass, plumule length, and diameter. As can be seen here on the right in these bubble photos, you can kind of see what our nursery setup looked like. So at the time of planting, propagules were randomly assigned to different treatments and tanks. And as you'll notice in the above little bubble there, soil and glass treatments were kept separate so that there was no nutrient crossover. In total, I had 8 tanks that were spanned across 3 different tables. Also, what you'll notice from the photo is that each tank was plumbed with a saltwater spigot. So through the entirety of the experiment, fresh salt water was flowing in as well as out of the tanks. I had 2 substrates, the crushed grass that we previously mentioned, as well as the potting soil. And I had 8 treatments total, 4 in the glass and 4 in the soil. And they looked a little something like this, right? So I had single propagules planted in a pot. I had 2 propagules planted together in a pot. And I also had a clumping treatment of a propagule and a wooden dowel control planted in a pot. We monitored growth for up to 6 months, and in that time, we collected different data such as plant height, number and length of leaves, leaf timing, number of branches, as well as salinity, water temperature, and air temperature data. For the water temperature, hobo loggers were placed in each tank, and for air temperature, we had a hobo logger that was fixed to the exterior of one of the tanks. As Miley Cyrus says, "It's about the climb." And indeed, she's not wrong. But for our purposes here, above ground growth is only half the story. At the end of 6 months, I sacrificed 160 plants based on nursery methods developed by Krause et al. 2003, thank you, Ken, and our awesome nursery intern, Zachary Briggs. Propagules were cleaned of substrate and then chopped into their bits, weighed, put in bags, and thrown in an oven for 8 to 16 days. And by doing this, we could evaporate all of the water out of the plant, thus leaving us with strictly mangrove matter, precious mangrove matter in our hands. And from that mangrove matter, we could look at biomass and thus look at a few things such as relative growth rates and absolute growth rates. All right, who's ready for some results? Yeah. Okay, So Miley Cyrus can buy her own flowers. She can hold her own hands, but she could not analyze my data for me. So unfortunately, I had to do it myself. And I began doing it by looking at those intake metrics that we had talked about earlier. So here we have the intake metrics all on one nice and pretty graph. And going from A to D and from left to right, we have mean mass, mean diameter or how fat a propagule was, mean total length, and mean plumule length. Now, the plumule is the soon to be newest set of leaves. And as you can see, and on the right here, it's just like that tiny little green sprig at the top of the propagule. From these data, we can actually sort of develop characteristics of the propagules as they were collected from each site, right? So here we see that Annenberg Bay and Perseverance propagules had greater mass and were fatter than propagules from Oasis Cove Marina and Steer. Additionally, we can see that Annenberg Bay propagules were the longest and that Perseverance and Steer propagules had greater mean plumule length. So their soon to be newest set of leaves was the longest. In addition to these metrics, I also looked at rooting tip condition. Rooting tip condition being exactly as it sounds, looking at the rooting tip of where roots start to grow on the propagules. Here in this chart, you'll notice rooting tip condition by site. And what's really interesting is that Perseverance has the greatest number of bumpy rooting tips, while Annenberg Bay and Steer have the greatest number of smooth rooting tips. Okay, all right. Well, while the literature doesn't say much about rooting tip condition, what they do say is that there are some signals here that growth later on could be accelerated by greater mass. So I thought, okay, well, if there's greater mass, maybe greater mass plus bumpy rooting tip condition can lead to more growth, ? And we would be partially right. A study by Ball in 2002 found that propagules with a greater initial mass grew more in 1 year than propagules with a smaller initial mass. Additionally, Lynn and Sternberg 1995 found that a few different metrics such as total plant height, carbon fixation rates, and biomass increased when plants were planted with a greater initial mass. However, pouty face, we have some limitations. So first of all, we have no idea how long these propagules have been on the ground, nor do we know how soon they begin to put out roots once they're on the ground. So we'll touch on this again later, but just keep that in your mind as we move forward. I just talked a little bit about what propagules looked like upon intake, and now we're going to talk a little bit about the environmental conditions that these plants grew up in over that 6-month period. So of the 480 plants that we planted, 472 survived. That's success, plant mom to the extreme. Of the 2 that died, of the 8 that died, 2 were in soil and 6 were in glass. You'll be happy to know that the average salinity throughout the entire experiment was 33 parts per thousand and was not found to be statistically different by tank over the entire experiment period. However, water temperature and air temperature say a little bit of a different story. So I did run statistical analysis of tank over the entire experiment period, and there were differences found. Some things did come back statistically significant. However, post-hoc tests found that things weren't significant. So I haven't been able to tease apart yet what is going on there. So I'm still looking at that data. Furthermore, air temperature was a little bit of a different story. We're here in the Caribbean. We know that as the year progresses, it gets hotter. Things get hot, the sun, she is in full force. So it is of no surprise that as we move from May, the start of my experiment, to October, the end of my experiment, we see that ambient air temperatures get hotter. However, as you'll notice on this chart on the right, this is a little snippet of my raw data. Right here, highlighted by the pink arrow, we have ambient air temperatures of 132 degrees Fahrenheit. Say what? Looking into this a bit further, this cannot possibly be true because this exceeds higher temperatures, even those known in Death Valley, which we know is called Death Valley for a reason. And so what we're seeing here is a possible albedo effect. And you know this effect even if you don't know it by its name, right? So basically this means that some things absorb more heat than others. If you go outside wearing a white shirt, you know that you're going to be much cooler than if you go outside wearing a black shirt, right? Because that black shirt, that darker color is going to absorb more heat. So it is potentially here that although our hobo loggers were small and green, that this green color actually absorbed more heat, thus messing with the overall temperatures it was collecting and not really showing us what was truly there. So more research has to be done on that as well. But nevertheless, we digress. If my clicker could work. Okay, so this is a wonderful scoreboard that Miley and I have put together for you as we go through our next results and graphs. And we're going to keep tally of who grew most where in each of these categories. Are you ready? All right, so let's start with mean total height here for the plants. Now, a two-way ANOVA found site and treatment, but not the interaction of the two to significantly affect mean total height. So we're going to look at those two things separately. But we're going to start with treatment. Here on the x-axis, we have treatment, and on the y-axis, we have mean total plant height. On the bottom here in the treatments, you will note that clumped propagules, two grown together in one pot, are averaged and thus only have one bar for that treatment pair, as noted by the DS1 and the DG1. Also, letters above the bars, no statistical similarities, right? So bars that have the same letters are more similar than bars that have different letters. When we think now to glass versus soil, soil versus glass, one of the first things we can know or note is that soil treatments, highlighted by the pink arrows, grew more and had a greater mean total height than glass treatments over on the right in orange. The lack of height here for the glass treatments is probably because of the lack of nutrients in the glass substrate. This is going to be something you're going to hear me say a lot. So good thing I'm not taking shots because I would be drunk by the end. Okay, so these plants don't have a lot of nutrients, but they're probably extra stressed because they're working hard. Mangroves are facultative halophytes, which is a fancy word to say that they don't need salt water to grow, right? They have those special adaptations, remember, that allow them to tolerate and handle such situations. So not only are they stressed because they have a lack of nutrients, I would be too, but also they're working extra hard to then exclude and excrete salt from the plant. Thinking now in terms of treatment, it is really interesting that all of the glass treatments are statistically similar to one another. When we move over to the soil treatments, we see that the soil treatment of the control propagule, right, the propagule planted with the dowel, has the greatest mean total height and grew the tallest of all the soil treatments. Over to the left of that, we have the single planted and the clumped propagules. These grew sort of similar to one another. And this is contrary to what I would have expected to see, right? I thought that clumped propagules were going to facilitate greater growth. You have two mangroves in one pot, that's twice as much mangrove power to grow upwards and outwards and outcompete everyone else. But in this case, not really. Moving on to site here, we have site on the bottom, and again, mean total height on the y-axis. And what sticks out like a mangrove sore thumb is that perseverance propagules have the greatest mean total height, coming in at 18.2 centimeters of all other sites, meaning propagules from perseverance grew the tallest. And we have our little steer propagules over here that grew the least of all the sites at 13.1 centimeters. Given all of that data, we're going to go ahead and give soil treatments, the control dowel and soil, as well as perseverance points on the board. Moving on to mean leaf area. The first thing I noticed about mean leaf area is the number of leaves that plants had. So the number of leaves for plants in glass ranged from 1 to 18, and the number of leaves for plants in soil ranged from 5 to 24. This same data can be seen down here on the chart where glass treatments are highlighted in blue and soil treatments are highlighted in red. And what I want to bring your attention to is the lower part of this chart, right? Where you can obviously see that soil treatments have many more leaves than glass treatments. This is important because the number of leaves as well as leaf length directly and indirectly relates to mean leaf area over time. So I ran a two-way ANOVA with mean leaf area looking at site and treatment. Site came back to be significantly influencing mean leaf area. However, again, post hoc tests showed no significance, so I can't tell you where those differences were. But we can't stop and we won't stop. So I changed up directions a little bit and instead ran a t-test looking at mean leaf area by soil and glass treatments via substrate. And what I found was that soil treatments had significantly more mean leaf area than glass treatments, meaning that soil plants had larger leaves. If you have a larger leaf, you're exposed to more sunlight. And if you get more sunlight, you're able to photosynthesize more, make more food, grow more, if you catch my drift. So from this, we are going to give soil treatments a point on the board. Now, we're going to be looking at mean leaf timing. Here, we have treatment on the x-axis, mean number of days to a leaf set on the y-axis. And please note that propagules that are clumped together are now plotted separately, and they each have their own bar on the graph. One of the first things we notice is that most of these treatments are statistically similar to one another, right? Except for the two blue and orange bars. So here we have a treatment that's different, and this is one of the glass propagules grown together in a clump. This one had the greatest mean number of days to a leaf set, about 30, while our little control dowel here in soil had the least number of mean days to a leaf set, meaning that control dowel propagules grown in soil produce leaf sets faster than all other treatments. Oops. There we go. Now we're going to look at mean leaf days to the site as well. And what you can see again is that our friend Perseverance here has the least number of mean days to a leaf set, meaning that propagules from Perseverance produce leaf sets faster than all other sites. Okay, interesting. Are we sensing a trend here? And one thing I should note is that I plotted site and treatment separately because my data did not meet assumptions for normal statistical tests. So I had to do two separate Kruskal-Wallis tests for these data. On the scoreboard, we are going to give control propagules and propagules from Perseverance a point. Branching out from our leaf timing data, 15% of plants had branches by the end of the experiment. And if we're thinking strictly in a soil versus glass setting here, soil plants had many more branch plants as well as many more branches on the plant. Try saying that, it's like 10 times fast. Woo, okay. All in all, glass treatments had fewer branch plants. And when they did have branches, there were fewer of them. We expect to see this, right? Because we already know that glass treatments are lacking in nutrients and probably overstressed compared to their nutrient-rich soil treatment compadres, which are just living their best lives. Looking at treatment now, an interesting trend develops. 28% of single planted plants in soil and 30% of clumped propagules in soil produce branch plants. But if you look, these numbers are similar to each other, right? Sort of similar to the other growth similarities we've been seeing between single planted plants in soil and clumped plants in soil. And of course, our control dowel in soil, again, it takes the mangrove cake and produces 43% of branched plants. Can you guess what I'm going to say about site? Yes, Liz has the answer, and you are right, Liz. And she said that Perseverance produced the greatest number of branched plants, coming in at 58%. Wowza. So what all of the data are telling me so far is that if you're a plant in a soil treatment, specifically with a control dowel, and you're from Perseverance, you not only have the energy and the means to grow upward, but you also have the energy and means to grow outward. And when we're trying to think about maximizing plant growth, this is an important concept to keep in mind. So again, we're going to give points to all the usual suspects here up on the board. But remember, above ground growth is only half the story. I sacrificed 160 of my plant babies to see what was going on below ground as well. And so the first thing we're going to do is look at relative growth rate, or RGR. Simply put, relative growth rate is looking at the biomass of a plant, right? That's strictly mangrove matter we can talk about at the end of an experiment minus the mangrove matter at the beginning of experiment, divided by the number of weeks in treatment. So you get a growth rate over time. Only a two-way NOVA only found treatment to be significant for mean RGR. So that's why we're only going to look at treatment here. On the x-axis, we have treatment. On the y-axis, we have mean RGR. And again, please note that on the x-axis, clumped propagules are averaged together. So you only have one bar for that pair. If you're saying, oh, Allison, you know what's interesting? This kind of looks like the mean total height graph we just saw earlier. You would be absolutely right. So, and similar to that graph, the first thing that we notice is that all our glass treatments are again statistically similar to one another and grew much less than all of our soil treatments on the left. Additionally, our soil treatments here were statistically different from one another. Although the single planted propagule here was not statistically different from the clumped propagules in pink or the control dowel in soil, the control dowel and the clumped propagules are different from each other. And that is because the clumped propagule in soil, the control propagule here in soil in green has the greatest mean RGR, meaning that it grew the most over time in terms of relative growth rate, while the clumped propagules grew the least over time in terms of relative growth rate. So we're going to give soil and control in soil another point. All right, we have one more growth metric to talk about here, and I hope that some of these categories can catch up because so far, it's looking like a doozy. I had a hard time trying to distinguish what the differences were between relative growth rate and absolute growth rate, but I think I have it figured out. So relative growth rates look at biomass at the beginning and the end of an experiment, while absolute growth rate just looks at biomass at the end of experiment. It just skips the whole initial biomass completely. So you're just looking at total absolute growth over time, which is in days rather than weeks. So just think of this as a different way to say total growth over time. And here, a Kruskal-Wallis test found only treatment to be significant, so we're only going to look at treatment. We have treatment on the bottom and mean absolute growth rate, or AGR on the y-axis. The first thing you'll notice is that, okay, dang, the glass treatments here are not only all statistically similar to one another, but they have much, much less AGR than all of the soil treatments. On the flip side of that, mangrove coin, all of the soil treatments have greater AGR and are statistically similar to one another. This is interesting because in previous graphs, we had seen that the soil treatments differed from one another and were more different, but here they're all statistically similar. The biggest takeaway from this is that whether you look at above ground growth or below ground growth, what absolute growth rate is telling you is that, hey, no matter what, soil treatments grew 4 times as much compared to glass treatments over time. So keep that in your brain. And because of that, soil treatments get the last point on the scoreboard. Okay, we made it through the graphs. Now the fun part, what does it all mean? I'm so glad you asked. Before we move on, I just want to say that no matter who scored points where and grew the most in whatever category, the important thing to note is that when you're determining what sites to collect from and what site and treatment combinations to use, you should always have project goals in mind, right? Project goals should absolutely run everything because results vary depending on what site and treatment combinations you use. Just as Miley Cyrus broke down her dance moves, we're going to break down the scoreboard in context of my hypotheses. So first, looking at soil versus glass here, I had hypothesized that soil treatments would produce greater growth than glass treatments. And as you can see, I was absolutely correct. Soil treatments wiped out glass treatments 5-0, okay? So plants grown in soil were taller. They had a greater number of branched plants. They had a greater number of leaves with greater mean area, etc. So if your goal is to maximize plant growth, then by all means, absolutely use soil as a substrate in your mangrove nursery. But remember what I just said about project goals, determining what sites to collect from and what site and treatment combinations to use. Don't discount glass just yet because mangroves were able to grow and crush glass. They did it. They were there. It's a little growth, but it is still something. So if you are a small island community with the means and time to repurpose glass in such a way, and you're trying to work with a minimum available of resources and maybe a more relaxed timeline for restoration, because plants that are more stressed will probably take longer to grow, then by all means, yes, use glass as a crushed glass as a substrate. It is an option. It is there. Oh, shoot, I didn't want to give it away. Okay, so there's a Miley Cyrus lyric that says, "The problem is all in your head," she said to me. "The answer is easy if you take it logically." And I'm happy to report that our team has been taking it logically, and we've already been exploring the use of a 50-50 mix of soil and crushed glass together in some smaller projects for some of our mangroves in the classroom work, where students get to run their own experiments and plant their own mangroves. However, future research should step this up a bit and do it on a larger scale, because this 50-50 mix could be great. You still get to incorporate a bit of sustainability while also capitalizing on all of the growth benefits of using the soil. I had originally hypothesized, remember, that the two propagules grown together would facilitate greater growth. And I did not see that, but it is seen in the literature. So as I mentioned at the beginning of this, plants clumped together can help each other out in stressful times, right? So in Florida, the Netherlands, when the same species of salt marsh grass is planted together, they help each other out biologically by sharing oxygen at the root level. They help each other out physically by helping prevent erosion at the clump edge. However, my results in this experiment vary depending on what site and treatment to use, so I can either accept or reject this hypothesis. But what I can say is that although propagules grown together did not facilitate as great of growth as I wanted to see, remember that they did grow on par sometimes in a few different growth metrics with the single soil propagules. So again, your project goals should suggest whether or not you use clumping as a treatment. Furthermore, I think it should be noted that upon further investigation, when I looked at propagules and plotted them separately for a few different growth metrics, I found often that one was growing more than the other. To me, suggesting that there's a little bit more competition than facilitation happening, right? So this also should be taken into account when deciding on your project goals. These same trends were most likely not seen in the glass treatments because, again, plants were already stressed. They already have their own thing going on. So they can't, grow everyone if they're trying to figure out how to stay alive themselves. So if you look up at the scoreboard at the right, which for those of you in the room is a little hidden, you can see that, okay, if you were looking at this, you would say, you know what? No, I'm using the dowel control propagule in soil, and that is my only preferred treatment method because it is the only one with points on the board. However, remember that wooden dowel was there only to say that there were benefits of structural support as well for the propagule growing with it. However, although the dowel did knock all the other treatments out on the scoreboard, it didn't rank the highest in all categories. And I'm absolutely not convinced that the physical support it provided was the only thing it was providing that propagule, right? I'm sure there was something else going on. And I have a few theories. The first theory is that maybe the propagule planted with the control rod had its mangrove senses tingling and said, oh my gosh, there's a potential competitor. I need to race to take up all the nutrients and space in the pot before he or she or it does. That's probably kind of maybe likely, but more likely what's going on is that although the control dowel looked intact at the time I threw it out for plant sacrificing, it probably decomposed a bit over those 6 months, leaching nutrients back into the soil substrate that plant could then take up for growth. This happens all the time in mangrove habitats in the wild, right? A tree dies, falls, decomposes, and then those nutrients provide growth to the living trees around it. Another theory I have, and I suggest this because I don't know anything about construction materials, is that perhaps there was some chemicals or some sort of glaze or something weird on the outside of the dowel that leached out into the soil. I don't know. Either way, these theories can either be confirmed or denied because they're outside of the scope of this experiment. So, of course, future research should look into plant competition in a nursery setting a bit more and explore the use of non-wooden controls. I would also offer the use of clumping more propagules together than just two, because perhaps there are benefits to clumping, but we couldn't see them because I only used 2 propagules together, and perhaps there are those benefits are there if there's more than 2 grown together. Okay, we talked about clumping, we talked about the control dowel, but we should not discontinue the use of a single propagule planted by itself. Because at the end of the experiment, they did grow pretty tall. And in fact, they had 6 to 8 leaves. And if you talk to other restoration practitioners, they'll say that this is substantial enough growth in a 6 months time frame to then outplant these mangroves into the wild. So although using the control dowel for some weird reason is a really great way to grow plants faster and have greater growth metrics, you have options as well as a mangrove nursery. Okay, so we talked a bit about treatment at length, but not so much about site. Remember, our biggest limitations in this study were that we have no idea how long propagules were on the ground. We have no idea how long or how soon they began to sprout roots once they are on the ground. And we have no idea how often propagules are dropped at different sites within the territory, right? So the reason why I had 4 sites in my experiment rather than these 6 is because of these limitations. Our team has been doing a really great job of studying the ecosystem function of health of mangroves in the territory over the last few years. And this research should continue, but should continue and include more phenology studies, right? So when are trees flowering? When are propagules ripe at each of these sites? This kind of data will definitely help us choose the best sites to collect from when those propagules are the ripest. In addition, we should also work smarter and harder, one of my favorite sayings, and include more replicates within our sites. I had a lot of plants. I had 4 sites. I thought I had enough replicates. But as some of my data showed, although initial statistical tests were significant, post hoc tests were not. And this is really upsetting to me because I did the work, I did the analysis, and to find nothing, that's a bit of a bummer. So upping the number of replicates per site could help tease out some of these differences and show where those differences actually are. In the meantime, though, we can continue collecting propagules that have bumpy rooting tips, because as my data show, those seem to facilitate quicker and faster growth in a nursery setting. We can also attribute the differences that we are seeing here in this presentation today, some characteristics of the collection site, right? Here in this photo, you have some of our wonderful team in the middle. Some black mangroves are in the foreground and the Bavoni landfill is in the background. It is no secret that steer mangroves are adjacent to the Bavoni landfill in St. Thomas. This landfill has been on consent decree to close since 2019 and has been cited multiple times for mismanagement of different pollutants and chemicals that certainly have leached their way into these mangrove habitats. Past MMES student Owen Clower did his master's thesis on this, and you should definitely check it out because some of the chemicals and pollutants he finds within the steer mangroves are pretty frightening. Similarly to this, Oasis Cove Marina is a mangrove habitat that is squeezed between a road and a marina. So there's no telling how often, how much, or what kinds of pollutants get into these mangroves and alter the ecosystem function and thus the propagule quality. A study by Zhang et al. 2006 found that a common oil lubricant, the author notes, that is found in subtropical and tropical regions actually stunts mangrove growth early on. So plants exposed to fresh or spent oil in this case had a less total height, less number of leaves, and less initial biomass. On the flip side, Annenberg Bay and Perseverance mangroves are more secluded sites away from human activity and thus a little bit more away from pollution that I'm aware of. So it's possible that because these sites are secluded, they have higher ecosystem functioning and thus greater quality propagules. As I mentioned, Owen's past work shed a lot of great light on the chemicals and pollutants that can be found in steer, but future studies should broaden that out to look at different mangrove habitats within the area. Last but not least, I had hypothesized that there would be different morphological differences of plants by treatment in site underground happening in the plant, right? And we found this when we looked at biomass. When we looked at the biomass comparisons in terms of relative growth rates and absolute growth rates, we found that soil treatments overall grew more than that glass, and that's fabulous. However, future studies should definitely include more replicates in this because I did look at plant morphology, the different types of roots and different root to shoot ratios more in depth, but I did not show these data here because I had a hard time from my low statistical power coming up with any results that were meaningful or being able to talk about things in a way that would make sense to you all. If you have questions later, please ask me. The takeaway is up the number of replicates. Although the techniques in practices used in today's study are somewhat novel, we are not the only ones championing mangrove restoration in the Caribbean. We are working alongside other people in the Dominican Republic, Puerto Rico, St. Lucia, and Anguilla, to name a few. And these pins you see here on this map, these are only the ones we know about. Because restoration happening on small island communities is happening on smaller scale, much of these practices and the restoration techniques are more locally known and missing in peer-reviewed literature and other literature as well. So what's great is that everything we've learned from our mangrove nurseries in inception in 2021, this study included, will go towards enhancing mangrove restoration literature and practices across the USVI and the Caribbean as a whole, which is great because we're not trying to reinvent the mangrove wheel. We just want the mangrove restoration wheel to be more efficient. Well, friends, we are nearing the end of today's presentation, and I want to thank you all in person and online, I see you, for your diligent attention. And we only have a few more things to say left, and this is what I really want you to take away from today's presentation. So please keep your ears, eyes, hearts, and minds open for just a little bit longer. The whole point of growing mangroves in a mangrove nursery is so that they can be outplanted later, helping restore our mangrove forests that have been devastated by continued deforestation and the 2017 hurricanes Irma and Maria. Unfortunately, growing mangroves in a nursery is not a direct indicator of success later on, because long-term monitoring and survival tracking are needed to see how many plants actually make it over a period of time. Are there obstacles that need to be overcome, such as drought or maybe some sargassum buildup? So although this part of my study has come to an end, my remaining plants will eventually be planted back out into the wild. Their survival and growth rates tracked over time, because in reality, we have zero idea how any of these plants are going to do, who will survive, who won't. I don't know, it's the Hunger Games of plants out there. It could be that glass treatments, already acclimated to stressful conditions, do just fine. And they say, hey, you know what, this is stressful, but we got it, and actually grow more once out in the wild. It could be that their nutrient-rich compadres say, oh my gosh, this is too stressful, I don't like it, and actually do poorer in the wild. We have no idea. What about Miley Cyrus? Well, Miley, she'll just keep being Miley. Does she actually know about mangroves or not? I have no idea. But if she did, if Miley did know about mangroves, if she knew how important they were to coastal communities like the US Virgin Islands, you can bet that she would be in here today with a glittery jacket, hand fist pumping in the air, tongue out, singing the praises of mangrove restoration alongside with me. And who knows, she might even throw in a couple song lyrics about mangroves saying, grow, baby, grow. I would not be standing here today if it weren't for all of the incredible funding sources and 2- and 4-legged friends you see up pictured here. I'm only here because of their financial, physical, and emotional support. So I cannot thank you all enough. These people and animals here helped me through the bad times, laughed with me through the good times, helped me with an incredible amount of data collection and management. They were my rocks, my awesome field crew, great mentors and leaders, and great family members as well.